To begin with some background, Naegleria fowleri, also known as the brain-eating amoeba, is a thermophilic protist that can be found in warm water habitats, which includes lakes in the tropics and hot springs in temperate zones. However, even though these are the most common places to find this amoeba, it has been found in samples taken from all over the world, including in places such as heated indoor pools, sewage, factory runoff, soil, and even some samples in tap water. In order to infect its hosts, it enters the human body through the nose while it's in the trophozoite phase, from which it attaches to the nasal mucosa, then moves along the olfactory nerve to reach the human brain. From there, it destroys the neurons in the central nervous system, attacks the meninges in the brain, and eventually attacks the brain itself. While considered a rare infection, it is one that has been recorded as occurring worldwide. The first case reported to the CDC occurred in 1962. Out of all infections which have been reported and documented, only 4 survivors of Naegleria fowleri infection have been recorded in North America, with only a total of 7 recorded as being cured worldwide. Once the infection has reached the brain, the amoeba will cause what is known as primary amoebic meningoencephalitis, or PAM. Once it has reached this stage, the patient will undergo inflammation of the brain, destruction of the brain's linings, cerebral edema, and eventually, in most cases as of present, death. The best chances one has of being cured of Naegleria fowleri is largely dependent on early detection and diagnosis of the amoeba. If left untreated, death of the patient will occur from brain death and edema within 1 to 12 days. While there are a good few symptoms that come with Naegleria fowleri infections, there is no singular symptom or set of symptoms which have been concretely associated with the amoeba, which makes diagnosis harder as some of the symptoms are indistinguishable from the onset of early bacterial meningitis. What symptoms do occur will usually appear within 2 to 8 days of the infection, but they can still occur in as little as 24 hours post-infection. Some of the most common symptoms are headache, fever, chills, photophobia, confusion, somnolence, seizures, and possibly coma.
Within its trophozoite form, its primary form of infection, the amoeba proceeds to invade the nasal mucosa, attaching itself to the olfactory epithelium. It then follows the olfactory cell axon utilizing the cribriform plate to the olfactory bulb. Once at the cranial floor, the amoeba begins to devour the brain and proliferate through means of asexual reproduction. Once it begins causing this damage, the tissue studied within patients via autopsy showed signs of hemorrhagic necrosis as well as inflammatory response, which implies that there is a distinct signaling cascade that occurs upon recognition of tissue damage. There was also an increase in neutrophils and eosinophils near sites of infection. The first cells of the immune system to encounter the pathogen are macrophages, neutrophils, and eosinophils. Macrophages are capable of phagocytizing the cell, but neutrophils are not. However, neutrophils and eosinophils are capable of successfully impairing the parasite using granules. There are mucosal-associated lymphoid tissues, known as MALTs, within the nasal passage that assist in forming the adaptive immune response as well as sample antigens for future use. The signaling cascade is triggered primarily by necrosis, and the tissue damage releases a high amount of DAMPs. These trigger PRRs such as TLRs and NLRs, toll-like receptors and nod-like receptors, which then activate APCs to present MHC class I to T cells. This then activates cell response using the MAP kinase pathways to initiate the said biological response. Meanwhile, another pathway is also activated via these receptors, which uses Cas1 to secrete self-IL-1 beta. This is the NLRP3 inflammasome pathway. Secretion of IL-1 beta is its response to receiving TLR signals and is a cytokine that plays a major role in cell proliferation along with inflammation leading to apoptosis, pyroptosis.
After APCs involved in the initial immune response have encountered Naegleria and are presenting it on their MHCs, they can activate T and B cells. Following activation, T cells secrete TNF-alpha, IL-6, and IFN-gamma, recruiting macrophages and neutrophils to the site of infection. B cells will receive help from APCs and helper T cells, and then they can begin to secrete antibodies. These antibodies will bind to the surface of Naegleria fowleri and further facilitate its clearance. As we will learn in the next slide, Naegleria has a way of avoiding clearance to the complement system. Once Naegleria reaches the brain, it will encounter microglial cells, which are a special type of macrophage that clears the CNS of dead and damaged neurons, as well as infection. When microglial cells encounter Naegleria, They will secrete cytokines such as TNF-alpha, IL-6, and IL-1 beta, as well as amoebocidal molecules, which will also aid in the eradication of this pathogen. When it comes to Naegleria fowleri, not all aspects of the immune response are effective in clearing this pathogen. The antibodies secreted by activated B cells bind to the surface of Naegleria fowleri. In most cases, this would result in the opsonization and clearance of a pathogen. However, Naegleria fowleri has a few tricks up its sleeve. Bound antibodies are internalized and capped, rendering them useless in aiding the initiation of the classical pathway of the complement system. Naegleria also has a CD59-like surface protein. CD59 is a MAC inhibitor surface protein and is crucial in preventing MAC attack on self cells. This CD59-like surface protein prevents the accumulation of the complement protein C9, thus inhibiting the formation of a pore and cell lysis. Cell-mediated immunity is the only effective way the body can clear Naegleria fowleri. Macrophages are large enough to phagocytose Naegleria. Neutrophils, on the other hand, are too small, but can kill this amoeba via degranulation of cytotoxic enzymes, the release of reactive oxygen and nitrogen species, as well as proteolytic enzymes. Neutrophils can also trap Naegleria fowleri through neutrophil extracellular traps, which are then cleared by macrophages.
Generally, Naegleria fowleri infections result in a 98% fatality rate. This means that there is a 2% rate of survival. Most doctors have never encountered the infection, and therefore, early detection and management of PAM can result in better clearance and resolution of the amoeba. The body can clear the infection via cell-mediated immunity. It just cannot clear it fast enough before it reaches the brain. In this sense, it can become a race between the immune system and the pathogen. However, in this case, the pathogen always wins due to the amount of brain tissue damage it can cause within the amount of time it enters the brain. Most of the research conducted to study N. fowleri has been through means of autopsy for humans. It kills the patient within 1 to 2 weeks of infection. Therefore, it is not possible for the body to defend itself alone. Survival can be slightly possible and can occur if the infection is detected early and treated aggressively within the proper environment.
As of the present, there exists no treatment for Naegleria fowleri that is considered satisfactory or effective by the wider medical community. However, there do exist some medications that, in conjunction with additional treatments, have been considered successful in treating the few known survivors of this infection. One of the primary drugs which have been used to treat Naegleria fowleri is amphotericin B. When administered, it disrupts the amoeba plasma membrane, attacking the ergosterol, and makes the Naegleria fowleri unable to form pseudopodia. However, even though it has been used and has succeeded in some treatments, it is a treatment that has very limited use, as it is toxic to mammals and will cause renal toxicity. However, in more recent years, miltefosine has also been used as a treatment for Naegleria fowleri patients, an amphiphilic phospholipid that is delivered to the patient orally, and has been often considered to be a new alternative to amphotericin B. However, these medications do not work entirely on their own, and are considered to only be effective when used in conjunction with other adjunctive treatments and therapies. Fluconazole is one such medication which has been used. It is an antifungal medication that, when used along amphotericin B, is believed to fight off infection in the body via recruitment of neutrophils. Additionally, azithromycin is another antibiotic which has been tested and recorded as being capable of suppressing over 90% of organismal growth. Rifampin is a medication which has been used in all surviving cases of Naegleria fowleri infection in North America. However, its actual role and ability in the successful treatment has been questioned in the medical community. In one of the patients, and something which has been possibly considered for future treatments, induced hypothermia has been utilized in order to help slow down the progress of patient death from Naegleria fowleri. Even though there are very few ways to detect and treat patients once they have contracted the amoeba, there are many ways to help prevent the infection altogether. One of the major ones being to avoid swimming or performing water-based activities in freshwater sources such as ponds, lakes, and rivers, particularly in the hotter summer months when the amoeba will proliferate more. If one still wishes to go into the water but wishes to avoid these freshwater sources, other sources such as the ocean or treated indoor pools are considered safe alternatives which will allow one to avoid infection. It is also recognized that there are certain medical and religious practices which are used which involve putting water in the nose, such as the rinsing of the sinuses, or, in Muslim tradition, the performing of ablutions before prayer. It is highly recommended by the CDC that one only use distilled, boiled, or filtered water.
In order to contract Naegleria fowleri, individuals must inhale it. This advice can easily be relayed to adults who understand how dangerous this pathogen is. The same cannot be said about children and adolescents. Another important factor to note is successful treatment depends on a quick diagnosis. In most of the articles I've read, scientists focused on treatments capable of penetrating the blood-brain barrier. I think one question that remains to be answered is how we can effectively prevent people from contracting Naegleria fowleri other than water avoidance. Traditional vaccines would be hard to develop considering this pathogen is almost always fatal. And as mentioned before, antibodies are ineffective against it. My idea is to investigate an intranasal cytokine prophylactic. In class, we have discussed administering lab-manufactured cytokines to boost the immune response against cancer. I think the same idea could be applied in this case. This nasal spray would contain TNF-alpha to stimulate neutrophil recruitment, as well as IFN-gamma to activate macrophages. The idea is to bring enough macrophages and neutrophil to the nasal mucosa so that if the individual inhales Naegleria fowleri, there's a strong enough immune response to facilitate its clearance long before it reaches the brain. Naegleria fowleri has been detected on every continent except for Antarctica, so everyone can benefit from receiving information about this pathogen, especially those who live near warm water sources. The social impact is very low because even though Naegleria fowleri is incredibly deadly, it is also very rare. In many developing countries, as well as the U.S., local warm bodies of water are used for cooling off, swimming, and in some cases bathing. The risk of contracting Naegleria fowleri has not deterred people from participating in these activities. As for the economic impact, I'll name a few. Measures taken to treat as well as avoid Naegleria fowleri can really add up. Antifungals are expensive and require medical supervision when administered due to their toxicity. Many countries do not have the manpower nor the resources to do this. A major way to avoid Naegleria fowleri exposure is to filter it out of the water. This requires expensive filtration systems. Not every country has the ability to provide these amenities for its citizens. When I lived in New Orleans for 5 years, we experienced frequent power outages due to the heavy rain. During the outage, power to the water treatment facilities was lost. Although there were backup generators, they didn't always kick in immediately, which allowed for unfiltered water that could potentially contain deadly pathogens like Naegleria fowleri into the water supply. During that time, all water had to be boiled before it could be used. Those of us who could afford to would buy bottled water and use that, but once again, not everyone is afforded that luxury. Research about Naegleria fowleri is being done all over the world. Aga Khan University in Pakistan, along with Georgetown University, are re-evaluating Naegleria fowleri and the new risk it poses to the people of Pakistan. This research is not funded and arose out of necessity due to the high rates of Naegleria fowleri in Pakistan, especially amongst males. Just last year, researchers at UC San Diego and Texas Tech published an article about combining cancer drugs with antidepressants as a possible treatment for Naegleria fowleri capable of crossing the blood-brain barrier. They received funding from NIH grants as well as the UC San Diego Startup Fund. Francine and Guy Cabral from Virginia Commonwealth University School of Medicine are researching the immune response in Naegleria fowleri. They received funding from the NIH.